The coming up speaker is an internationally renowned and acclaimed researcher, a physiologist as well as a department head, and she has been appointed distinguished professor at Stellenbosch University. She's worked on conditions such as type 2 diabetes, rheumatism, Parkinson's and Alzheimer's disease, but her contributions to research on long COVID and ME have led to a better understanding of lingering symptoms in these patients. So all the way from South Africa, we're honored to have you here. Please join me in welcoming Professor Resia Pretorius. Maybe.
Yeah, that's correct.
Thank you.
Hello, everyone. It is a massive opportunity for me to be here, and I am so happy to meet everyone. We've had a wonderful roundtable yesterday with lots of new collaborations going, and it's a pleasure for me to just present our research here today. This is just some of my collaborators that I wish to acknowledge. There are many, many more. These are mainly my students from my South African lab, but then also researchers that the work that I'm going to present today have had a significant contribution, particularly Professor Douglas Kell, my long-standing collaborator from University of Liverpool, and then a few other people that I would like to recognize. And also the funding that I have received over the years, Polybio Research Foundation, Balvi Research Foundation, Standing Up to POTS is our latest grant, and many more. So I just wish to acknowledge them as well. So if we start with long COVID, although the general community, governments, as well as even some clinicians, still act as if the pandemic is now long gone and over, that is not the case, and it's definitely not true. If we look at numbers, 400 million people globally are suffering from long COVID. 10% conservatively, but up to 30% of individuals living with COVID and having had COVID would have gone on to become long COVID sufferers. If we look at the economic burden, it's realized that it's going to be around about $1 trillion per year, every year, for the US alone. If we look at excess mortality rates, it's up to 3% higher than pre-pandemic levels. And it's believed that these numbers will stay with us till around about 2033. That's a massive lot of people dying due to excess and due to excess mortality rates that we see. Now, if we think that long COVID is a global health crisis that isn't over yet, we have totally been forgetting that ME/CFS is a health crisis that has never been taken seriously by governments, by some clinicians, not all, and it's extremely understudied and underlooked at patient population, the left behinds. If we look at all of these numbers, it is a large amount of people suffering from ME/CFS. And if we take into account that many people suffering from long COVID will progress into ME/CFS, it is even more striking, and the numbers are just going to be on the increase. So let's look at what we have been saying and our colleagues have been saying on, if we first look at long COVID, what are the background, the hypotheses driving long COVID? So obviously I'm working in clotting pathology, and we discovered microclots and their effects in this disease. But if you look at long COVID, you can see that microclotting and endothelial dysfunction that we just heard about in the previous talk, all of those can be traced back somehow to immune dysfunction, to cytokine dysregulation, to muscle pathologies, and also viral persistence. And if we take into consideration all of these different possible avenues that are involved in the development and the disease symptomology of long COVID, we can all trace them back to an endothelial dysfunction and also a thrombotic endotheliitis. So a thrombotic, meaning clotting pathology, related directly to endothelial dysfunction. Now, what are we saying and what are we not saying? We are not saying that every single individual with long COVID might have microclotting or thrombotic endotheliitis. We are not saying that, just as we can't say that it is a homogeneous disease. We know that long COVID has got many symptoms, and in a recent publication, we also noted that there are possibly different types of long COVID patients. So we need to figure out which patients have got these type of pathologies. How can we group them together? So it's not a one-size-fits-all diagnosis, nor one-size-fits-all treatment. If we circle back to ME/CFS, in 2 recent publications that we have done, we have also discussed the hypothesis that ME/CFS and endothelial dysfunction and the symptoms that we have noted in ME/CFS can all be brought back to viral reservoirs, possibly, nervous symptom effects and symptomology, immune cells, as we all know the disease and have talked about it. But then, if we look at all of those together, endothelial dysfunction central to the disease pathology. And we think, linking to that, we have clotting pathology as a possibility for the pathology in the condition. We have also published a paper recently where we looked at herpes virus infections and endothelial damage in ME/CFS. We note that endothelial cells might have a significant role to play. We think that these herpes virus, and we know especially from other researchers that they can indeed infect endothelial cells. We also know that there might be long-term latency and persistence involved, and that may lead to and drive a clotting pathology in this condition. And flow reduction, blood flow, and as Ingrid pointed out previously, damage to the endothelial function might occur directly as a result of these herpes virus infections. And then here is just one of our figures in that publication where we try to bring everything together: autoimmunity, neuroinflammation, vascular dysfunction, even platelet hyperactivation. All of these different parts of what we see in publications that some people have been writing about this, and our own observations lead to widespread endothelial damage and clotting pathologies also in ME/CFS. Now, if I circle back to what we have been doing in our lab, we have been looking at endothelial cells, their receptors, how they interact with different inflammatory molecules that might be in circulation. Endothelial cells have got numerous receptors. These receptors sit on the cells. They may interact with any possible inflammatory molecule that comes past. We say it's like a lock and a key effect, where the lock and key, they interact with each other, they turn on something. The endothelial cells react to this turning on of an inflammatory molecule, reacting to it. It can damage it, and it can produce all sorts of inflammatory molecules again from these damaged layers. And as Professor here in front mentioned earlier in the questions, there's like it's a huge area, surface area of damage if you've got a general endotheliitis or inflammation of your vasculature. It's a huge area, and it can cause significant damage to your body, to your symptoms, to your organs, because this is the way how oxygen is transported in your body to your organs, to your brain. Remember, the blood-brain barrier is nothing else but an endothelial layer preventing stuff from the body reaching, penetrating and going into the brain. So the endothelial layers of this blood-brain barrier is compromised. You get things into your brain that's not supposed to be there, inflammatory molecules that can damage your brain. We have been looking at those endothelial layers. Then we've also been looking at platelet activation. Now, platelets, small round balls, they're supposed to just float around, and they activate when you have you need to clot. If you cut yourself, platelets rush first to the area. they get hyperactivated when they interact and get in touch with all sorts of inflammatory molecules. And then fibrinogen is a soluble clotting protein that we are interested in that can turn pathological in the presence of inflammatory molecules that might have a protein-protein interaction. Now, we have been looking at all of these in different inflammatory conditions, and then when COVID came along, this was our focus as well. Quite early on, we decided that we should look at what happens when you have got a problem with oxygen transport to your cells, to your organs. What happens is you get ischemia and reperfusion injury. So the ischemia part is where you don't have oxygen and then suddenly you have a rush of oxygen to the area. Oxidative stress happens, damage to the area, and then you have sometimes also have a reperfusion when the oxygen gets reperfused into the cells. We think that's a process that should be looked at carefully in long COVID as well as ME/CFS. Now, quite a few years back already, probably since 2011 to 2014, 2016, we were looking at the clotting protein fibrinogen. It is supposed to be a soluble protein until you cut yourself, and then it becomes a little gel. Fibrinogen goes to fibrin to make a little clot. We have realized quite early on that in the presence of inflammatory molecules, the inflammatory molecule might directly bind to the fibrinogen and change the biochemical structure of the fibrinogen molecule. So it untwists from little coils, alpha coils, it untwists and forms beta sheets. And this forms a little insoluble entity, particle, or dense matter deposit, if you would like to call it that. Previously, what we did, we took plasma from healthy individuals and we added thrombin to it to cause a fibrin net. We used thrombin to activate it into fibers. And here's quite a nice experiment that we did in 2016, where we took these plasma from donors, from healthy donors. We added theoflavin T that marks amyloid misfolding in proteins. Amyloid, this protein marker was used for amyloid in the brains, but it's not the same. Beta protein in the brains of Alzheimer's can also be marked with theoflavin T. That's where it's been used for a very long time. But it marks open hydrophobic areas on damaged protein. So in sample A, you can see we added thrombin, nothing, no amyloid parts. Then in B, we added LPS, and that comes from the outside layers of bacteria. It's a well-known inflammatory molecule. If we added LPS to plasma and then our marker, the green marker THT, it induced these changes to the plasma protein fibrinogen, and it caused it to become amyloid in nature. And then, in fact, we could also reverse it by adding this LPS binding protein that binds LPS and remove its action. And then we could remove the signal that we saw. So that was the type of things that we did previously. We always added thrombin. And then we started thinking, well, let's see if we can also add thrombin and add other inflammatory molecules to cause changes. And here's an example where we use amitracker, which is a marker similar to theoflavin T. It's just another one that marks amyloid protein. And that in this case is the red signal, and the green one is serum amyloid A, which is an inflammatory molecule that sometimes is in circulation. And we saw that if we add this inflammatory molecule into our plasma, or into fibrinogen, purified fibrinogen, we could also induce these amyloidogenic changes. And here's an example where we added we looked at fibrinogen, but in this case, the fibrinogen itself was green. You buy it, it's green, and then you add serum amyloid, and you could see these changes to the structure of the fibrinogen. And in this colorful one, we'd also added serum amyloid A to healthy blood. There you could see red blood cells and platelets at the bottom, hyperactivating, and the red blood cell gluing together. So that gave us an indication that we could also induce these clotting pathologies. So when COVID came along, we looked at healthy plasma, diabetes plasma, and COVID plasma, but at this stage, we couldn't find thrombin from our blood bank that provided it for us because of the COVID pandemic. And lo and behold, we saw that in COVID, we have got these large amyloid clots forming without even inducing clotting using thrombin. Where in healthy plasma, very little is seen. In diabetes, we do see, because we know diabetes is also a disease with clotting pathology. Then came along long COVID, and we did the same experiment. We saw that we have still got significant microclot formation at the top in platelet-poor plasma. So all the cells were removed, only the plasma was left, theoflavin TR marker was added, and we still see clotting pathology in these patients. At the bottom, the scanning electron microscope photos is where it's just blood from a patient, from a long COVID patient, onto a slide under a scanning electron microscope. Look at all the microclot formations, these dense matted deposits of the plasma protein that clogs up and around the red blood cells. Now you can just imagine those red blood cells flowing in your circulation, trying to take oxygen to all of your organ systems. They won't flow properly. They won't be able to squeeze through the capillaries. Remember, a red blood cell's got this strange little indented half-moon shape indentation in the center. They are prone to and they should squeeze through fine little capillaries. They won't be able to do that if they are glued together. They won't be able to get oxygen to the cells. Remember the ischemia reperfusion injury and endothelial damage associated with that? Red blood cells aren't functioning properly in long COVID and in COVID. And here you could see just an example of one of these microclots. This is a Z-stack in a confocal microscope, just showing the extent of the size. This was a very, very ill patient, actually a clinician, that I looked at his blood samples in Germany in 2021. So the question that we get, so what does it matter? We see microclots in various diseases. Does it matter? Does it matter that they are there? Do we need to have any attention to that? And the question that I also get is, are they indeed clots? How can you say it is a microclot? How is it related to macroclots causing stroke, causing heart attacks, deep vein thrombosis perhaps? And to answer it, the first question that I posed, should we pay attention to it? Yes, we should, because it's a novel marker and a novel entity that might describe better what we perhaps in the past couldn't describe. Perhaps it's a place to look for novel inflammatory molecules entrapped inside these clots. Now, so the question is it a clot? Should we call it a clot? And the answer is yes, definitely, because if we look at the definition that the hematologists and the researchers and clinicians working in clotting, what they give us as a definition. First of all, they say, inside a clot, a thrombus that causes stroke, heart attack, you will find fibrinogen. Okay, so that's a good marker. We need to then determine whether fibrinogen is in the clot. Is the protein that we have found that turned amyloid fibrinogen? That's a question. We agree. Then also, red blood cells, white blood cells are entrapped in these thrombus, some macroclots. So if I say that what we see are microclots, they're not yet these massive clots that engulf lots of red blood cells, but I would suggest that seeing them on the outside of the red blood cells as in the previous scanning electron microscope photo, that indeed they are precursors of further clotting pathology. So the other question that I had actually yesterday was a very valid question. So why don't we see strokes and heart attacks left, right, and center in ME/CFS and long COVID? And that is a valid question. But the answer to that possibly can be many diseases have underlying clotting pathology. Not all of that lead to a full-blown event like a heart attack or a stroke. So, and indeed, there are publications that now say that in long COVID, you have an increased risk of both stroke and heart attack. So to look further on what are the content, so to clarify if these are microclots. In 2021, we looked at long COVID and acute COVID, diabetes, and control samples using proteomics. What we did is we took the plasma when we wanted to search for molecules, for protein footprints in the protein, the plasma. To our surprise, we could not digest the acute COVID and the long COVID using a typical enzymatic process that you put enzymes to digest for your mass spec methodologies. It's bog standard methods, but we couldn't digest the acute COVID and long COVID. We rushed back to the lab, added our theoflavin T, and we saw after a first digestion, all the clots were digested in controls, very few little clots that they are. Diabetes also, we couldn't find any evidence that there were anything left. But indeed, in the long COVID and acute COVID, the clots were still present. Then we followed a process with our biochemists at the unit, the proteomics unit. We developed a double trypsin digestion method. Then we could digest the clots, and lo and behold, there are the various molecules that we found. Now, inside, in the entrapped clots that were now broken open. Obviously, as we thought, we would find all the different chains of fibrinogen. Coming back to the question, is it a clot? Yes, fibrinogen is present. We also found von Willebrand factor, serum amyloid A, that a few years ago we saw can cause microclot formation or structural changes. We also found something interesting, alpha-2 antiplasmin. Now that molecule prevents clot breakdown. So now for the first time, we had evidence why in long COVID and acute COVID, alpha-2 antiplasmin, when it's present, prevent the clot from a regular breakdown by your body's own processes, the lytic process, the fibrinolytic process. If you've got a clot, your body's got the potential to break it down by this fibrinolytic process. But if you've got molecules that prevent clot breakdown, that blocks the plasmin process, the clots may take longer to break down. So recently we also took, again, purified fibrinogen. We added to it spike protein. We did it in a previous experiment in 2021 too, and I'll show you that in a moment. We added, the red here is the alpha chain antibody. So we added spike. It caused a little microclot to form. We then marked the fibrinogen with this alpha chain antibody. And we also added theoflavin T, which is our marker for amyloid protein. And if you look at the two and the overlays here, you could see that areas of this microclot does indeed contain a significant misfolding or amyloid protein. We also did the same by adding LPS. So previously in 2016, we did it by adding LPS and thrombin. Now we just added LPS. And here you could see the overlay. The LPS causes significant areas of the microclot to change and become amyloid. In 2021, we did an experiment, a microfluidics flow experiment. And there you can see, if we added, first of all on that side, you could see if you add spike protein, it changes the structure of the red blood cells, the microclots appear. And in this, at the top of the nearest tube, you see healthy plasma with little clots forming. COVID sample, huge clots in the plasma. And at the bottom is spike protein added to the same healthy participant as above. So spike protein on its own can cause significant microclot formation, and this is under flow. We simulated a blood vessel with this system. Now recently, Rayo et al. Published a very lovely paper that I'm so glad that I saw this. In Nature, where they said that there are pathological changes to fibrinogen in the presence of spike protein. We also had a collaboration with Professor Alain Thierry from INSERM in France. He's an expert in netosis and amyloperoxidase, which is a protein that is produced and present on nets, which is an indication of active inflammation in the body trying to get rid of it. And we at the top there, we did some machine learning of these microclots. And you could see there the red in the middle. I don't think this cursor works, but the red in the middle is the nets marked with cyto. Cyto is a red marker for nets. And you could see the two little clot parts, the fibrinogen amyloid clot in both sides and the net entrapped in between these two areas. So what was here a confirmation to us that netosis and the products of it is central in long COVID as well. And we also did myeloperoxidase, elastase, and also different markers for netosis the team did in their France laboratory, and they did found significant changes between healthy and long COVID. And then here is where in our lab we did again the Z-stack. We used theoflavin T as an indication of our amyloid protein, fibrinogen, the red one, for our fibrin. And then the MPO, the blue little marks there, is for the myeloperoxidase. Now, we don't have the red marker here for the net, or you could have used Hochzat as well to show the net. But you can imagine that there's an imaginary net that we didn't mark here that extends out of this clot, and the MPO is sitting at different parts of this little clot. We were very, very happy to also have a collaboration with the VUS lab from Amsterdam. A few months ago, they published in Nature Communications where they used our methods, theoflavin T. They did biopsies of long COVID during and after, and a few days further along after patients were on a treadmill for post-exertional malaise. And they found significant microclotting inside the muscle of the patients with long COVID. They just had a repeat experiment with ME/CFS as well. Our collaborators in Sheffield Hallam could mimic our results as well. They've got a ongoing long COVID study, and they have got uninfected controls that had COVID but now recovered, long COVID, and recent COVID within a few months after a COVID infection. And they used our methodologies with another instrument, and they could repeat it. We were also very happy to see that in a pediatric group from our Harvard collaborators from the Lyle group found nets associated with microclots in a pediatric population using our methods as well. And then we were very excited for the Toe group in University of Liverpool. Professor Toe is a very well-known emergency medical doctor and clinician, and his group had stored samples from sepsis individuals. And they used our methods, confirmed that there are microclots in sepsis as well, that there are indeed clots because they also found the fibrin nets, and they found the exact same molecules as we did, inflammatory molecules entrapped inside sepsis patients' blood samples using proteomics. Now, what about ME/CFS? A recent study that we have completed and published on showed that in ME/CFS we've got significant platelet hyperactivation. We do have microclots, but not to the extent that we see in patients with long COVID. These samples were from patients who never was exposed to an acute infection. We also did a proteomic study with these samples. And we also found different footprints of inflammatory molecules, but also clotting pathology markers entrapped inside these microclots. So obviously, proteomics is a very difficult method. We can't do it per patient. It's expensive and it takes a long time. Microscopy is tedious. Not many patients have access to research facilities that's got fluorescent microscopes or confocal microscopes. So in 2021, we crowdfunded together with Balvi Foundation and Polybio Research Foundation, and we got an imaging flow cytometer in our lab. And we published our first paper to have a method to show that you can use an imaging flow cytometer, or for that matter, any flow cytometer. You can use, and you usually use a flow cytometer to look at cells, but we pushed through platelet-poor plasma, so cell-free, and we could find these microclots with our theoflavin T as our marker, the green marker, and we could compare levels between controls and long COVID. And here's just an example of how the differences between the two groups look like. We recently had an imaging flow analysis of our ME/CFS controls in long COVID, and here you could see that there's definitely a significant difference between the clots that we found in ME/CFS versus long COVID. So on the disease pathology, what is the difference? Obviously, with long COVID, spike is central here again. We also then thought, okay, let's look at size ranges. There are different size ranges of clots. How does it compare? So it seems to us as if in a healthy individual, you do have clots, but they're in the smaller size range, and probably they flow easy through your body. The dilemma comes when you've got the biggest clots, and I'm talking about 1,600 plus micrometer in diameter. They are squishy little entities. They can maneuver through the capillaries. But some of these patients have got significant loads of these larger sizes. Then we talked about endothelial damage. We also used our imaging methods, our microscope, to look at endothelial debris in plasma. And here you could see that in controls we hardly see endothelial debris, and this is a marker that is called E-selectin, which is a very well-known damage marker for endothelial layers. And we do see in long COVID some endothelial debris, and in ME/CFS this huge long piece looks as if it's shredded off from the inside layers. We were very happy to see a publication of some of our collaborators, Michael Peluso, as well as Steve Deeks. They just published a week or so ago, where they said in long COVID, microvascular dysfunction and tissue hypoxia should be central in treatment regimes and diagnostics. And then here's the mechanism for targets for long COVID. Once again, central. Microclotting. And this is so exciting for us because we seem to have getting more and more collaborators seeing what we have seen, doing our methodologies that we developed. So what's the next steps? Microclot testing, we believe, is a new diagnostic frontier. We have worked with everyone that wants to work with us. We have now got collaboration agreements with people here in Europe. Innes Laboratories has got an imaging flow cytometer. They will be doing microclot testing very soon. We've got collaborators in Germany that's in different clinics that are already doing this. We've got collaborators in the UK, and in the US there's clinics using microclots as a measure, a diagnostic measure. Then recently, about a week ago, we just published this paper where we said, what are the methods that can be used in the absence of microclot testing? Because it's not in laboratories everywhere. So what do we do in the meantime? So things like FMD that Ingrid talked about, radiology, even nail video capillaroscopy, those are things that can be used to show that there are some sort of damage. So we have got numerous collaboration, but one of the exciting ones that we are busy with is a collaboration with the LINK study, the NIH study, where Michael Peluso, who's got this NIH grant, he's been looking at post-acute COVID research programs that he's been doing, and they are evaluating the spike antigen-induced coagulopathy, or clotting, and neuroinflammation. Now, the RAIO publication that I talked about, they are included in this collaboration, and we are very happy to say that we will be receiving samples from this study. Unfortunately, not funded by the NIH. We've got PolyBio funding to do that. They will ship all their samples to my lab. So we will be doing the microclot test in our lab for this study. And we've got numerous other collaborations, and I hope new collaborations that will start soon coming from this meeting. Now, the take-home message is, if we look at microclots, it's not a question the size or the numbers. We should not be saying it's present in all diseases, so what? It's much more than sizes and numbers. It's their content. It's a new frontier. It's a new marker. We haven't been looking at platelet-poor plasma to see things in it using flow cytometry. It was hiding in plain sight. It's a novel entity that we must now figure out. We don't know much about it, and definitely our group don't say that we do. We need to take hands with everyone interested in biomarker searches, see what we can find inside them, and see if their activity is important for us, and see if their biochemical characteristics are important, and perhaps it can be a useful instrument as a diagnostic tool before and after a clinical trial. Measure before, measure after. Also, it could be if we've got 5,000 long COVID patients or ME/CFS patients in front of you, you know that each one would not have exactly the same pathology as the next one. At least we could use this microclots as a possible mechanism to group patients into treatment groups. That might be a possibility. So the possibilities are endless. And I hope I've convinced you that we must look closer at endothelial damage, microclotting, and their content for diagnostic markers, not only for ME/CFS and long COVID, but for all disease types. I thank you.
Thank you so much. Thank you so much, Dr. Pretorius. I'm sure there are questions for you too. Who has a question for Dr. Pretorius? Can't see anyone. Over there, please.
Thank you for an interesting talk. You present data from blood. I wonder if you have any data from tissue or in vivo experiments that support your ideas.
Sorry, I missed the first. If I have blood, if I've got samples from?
If you have any, I wonder if you have any data or studies from tissue.
We don't have studies from tissue, but the only studies that there are from tissue is from the Wust group and from Amsterdam. They have taken muscle biopsies and they have found microclots just on the inside of the muscle next to endothelial layers. So what their hypothesis is that in individuals with long COVID and ME/CFS, the endothelial layers are very much damaged, and somehow the microclots in circulation can move into the muscle and go and sit in the muscle and cause pathology. So they are working now on another study. They've done a first study on long COVID. They're now finalizing their tissue-related study to expanding it to ME/CFS. And what I do know is they've got funding to actually take these biopsies of their muscles that they have and do transmission electron microscopy to actually see the interactions with the microclots in the muscle and or on the vasculature. So I'm waiting for those results. It would be quite interesting to see that.
Is that a kind of in vivo model to test your to support your findings?
Well, I suppose it could be, yes. I think it's a good model to use. It would be very interesting to see other types of microclots in other parts. We've applied for a funding initiative together with another group, Jeroen den Dunnen's group. We are planning to do PET scans of brains in long COVID and perhaps also CFS versus controls to see if we could actually find microclots inside the brains using PET scans. And we've got a whole team of experts in PET scans.
Thank you for information.
Extremely interesting as everything that you have been speaking about. Thank you so much. Any more questions in the room? In the middle.
I'm wondering your thoughts about the lack of oxygen to muscle and to the organs. Do you think it's more cell death or chronic conditions to this state, or is it reversible? Does it cure itself if we find a reason, or is it more cell death and organ damage, you think?
I think it starts off with widespread vascular damage. So systemic vascular damage, red blood cells not being able to travel into the vascular bed near different organs, and they being unable to get oxygen to the different systems, the organs that they need to give oxygen to, and also, in fact, remove CO2 after the cells have used it. And so widespread endothelial damage, oxygen depletion, this ischemia, reperfusion injury, post-exertional malaise might be related to oxygen not being traveling into the muscle after a event of a walking event or exercise event. And so cell death, yes, might eventually happen, but I think there might be a possibility if we could return the endothelial layers to its health status, we get the clotting to be normalized, and we possibly remove the inflammatory onslaught that is causing it. So in those, along those lines, we're thinking what many researchers have been talking about, viral persistence and a reactivation of latent viruses, herpes. If we can remove that by perhaps Paxlovid or any other type of medication that I know many people are trialing, that might be an idea to remove the inflammatory onslaught and then heal the endothelial layers and let the coagulation normalize. I think that would be the way. So I don't think it's necessarily a dying off of cells in such a way that it's destroyed, but definitely cellular death might be an important thing to look at.
Thank you.
Thank you. Lisa in the front.
Well, it seems like we have to do with a culprit that is not invisible but hard to see. And you mentioned a few alternative proxy investigations. But what do you think about retinal photography? Could that be of use?
Absolutely, that's an excellent question. So retinal tomography, you look at the blood flow in the eyes, specialized mechanism, but I do know some clinicians that have been using such a model to look at the vasculature in the eye, do see that they are compromised, sometimes rarefication of the endothelial layers. You also see rarefication in the nail fold. So what it means is a reduced blood flow and formation of the little blood vessels in the nail fold, and with the retina, some clinicians have also been noting that. So it's perhaps not so accessible method to look for it, but at least it can show you that you've got some issues in your little fine vasculature in your eyes as well. And many patients have been complaining of eye effects.
Thanks.
In the front.
Do you think there's a way to accelerate the normalization of the microclotting? particular approaches to do so? Because I noticed the difference in high microclots in long COVID or recent COVID, fewer in ME, and then fewer in healthy.
Yeah, that's a very controversial question, and I think it needs lots of clinical trials and further investigation. Some clinicians that I've been working with have been using a triple anticoagulation therapy to reduce the clot load by preventing the clots to form and to calm down the platelets, so that the endothelial layers have got a chance to respond, to heal. Now, if I mention this, many clinicians' hair will stand upright, because the first thing that you think about is massive bleeding risk. But in the right hands, if the clinician has got the right tools to show that there is a clotting pathology, there is platelet hyperactivation, we have seen that possibly around about 80% of the individuals that's taking these medication within our local clinic where our clinician uses the treatment have felt better. So felt better, I'm not saying return to full health 100%, but they were on their way to feeling much better. So on their way to recovery. That said, 20% didn't respond. The bleeding risk is significant, but we didn't find bleeding because if the coagulation is dysregulated and the platelets are hyperactivated, you're just normalizing everything, so there will not necessarily be a bleeding risk. Similar bleeding risk possibly then for someone with a stent after a heart attack, which also clinicians also give these type of medications. Now, there are some easier routes via nutraceuticals. There are products called nattokinase and serrapeptase. They are enzymes without a bleeding risk. Many patients have reported that when they take these medications, ME, CFS, as well as long COVID, that some of these symptoms are reduced. Now, what happens is those enzymes don't block clotting fully. They break down clots, so you don't have a bleeding risk. Nattokinase is in natto, the Japanese food source that many Japanese individuals eat, and we all know they've got lower risk of cardiovascular disease. Might be to the natto that they take in their diet. Serrapeptase is a product that breaks down biofilm, which now circles back to herpes virus and other bacteria that might be involved in the diseases. But it seems as if those more kinder to your blood coagulation system have worked very well for many patients, and we've had wonderful reports from people feeling a little bit better on those type of treatments.
There seems to be some hope, at least. I'm so happy for that. Thank you so much. Give a big hand to Professor Pretorius.
Thank you.